My name is, thanks, good on. My name is Nicole Gordon and I'm the CEO of Genetics, and I'm delighted to welcome so many of you to our latest talk in the Diving Into Our Gene Pool series, which is called Cracking the Code of Crohn's. Obviously a very emotive subject, probably the largest numbers we have ever seen on any of our Diving Into the Gene Pool series. So welcome, lots of new faces in the room this evening. So a little bit about genetics. We exist to raise awareness, educate, and to test the community for Jewish genetic disorders. JGD is a term that we often use to describe genetic conditions that are more common in people of Jewish ancestry, and that's including people from Ashkenazi, Sephardi, and Mizrahi backgrounds. Genetics now tests for 49 of the most severe recessive JGDs, all of which have no known cure. Since 2014, we have screened close to 9,000 individuals in schools, universities, and through our genetics clinic, educating tens of thousands of young people and identifying numerous carrier couples who we have helped to go on and have healthy families. You probably recently heard a lot about the NHS BRCA testing programme, and we've been working really closely with NHS England as the official engagement partner for this groundbreaking programme, and that's alongside High Cancer Care. Since launching the programme, an astounding 43,000 people have signed up to be tested through this first of its kind groundbreaking programme, and over 500 BRCA carriers have been identified. BRCA gene faults are at least 6 times more common in those with Jewish ancestry, aren't we lucky, compared to the general population, emphasising the importance to our community of this fantastic programme. Both the recessive screening programme and the new Jewish BRCA testing programme give people the knowledge and the power to make the best decisions for themselves, their future families, and also the whole of our community. For more information about either of these tests, please visit our website, and hopefully someone's going to put our website into the chat. As well as genes coding for recessive disorders are leading to a predisposition to cancers, we are also aware of a genetic predisposition for conditions affecting the digestive system, such as Crohn's and colitis. Anecdotally, as members of the Jewish community, we likely all know somebody with one of these conditions, and really this demonstrates the whole interaction between health and ethnicity. Tonight we will be hearing about the genetics behind Crohn's disease and how living with Crohn's affects so many. If you have questions during the presentation, please feel free to put them into the chat and we'll get them to at the end of the talk. So tonight we're delighted to welcome James Lee. James Lee is a clinical scientist group leader at the Francis Crick Institute, a professor of basic and translational gastroenterology at UCL, and a consultant gastroenterologist at the Royal Free Hospital. He trained in medicine at the University of Oxford and gained his PhD from the University in Cambridge in 2011 as part of the inaugural Wellcome Trust Clinical PhD programme. Professor Lee moved to the Crick in 2021, where he now leads the Genetic Mechanisms of Disease Laboratory, seeking to translate genetic associations into a better understanding of autoimmune and inflammatory disease biology. Now I'm delighted to welcome Professor Lee. Over to you.
Thank you very much, Nicole. Thank you for everyone for joining this evening. So as Nicole has just outlined, my name is James Lee, and I principally lead a research group at the Francis Crick Institute, which is this enormous biomedical research institute directly opposite St Pancras in the centre of London. And the work I'm going to talk about today has really been led by 2 people in the lab, but actually all of the other people shown with blue outline here are also working on aspects of Crohn's disease and ulcerative colitis genetics. The other thing that's relevant here is that I'm also a practicing gastroenterologist, and I look after patients with inflammatory bowel disease up at the Royal Free Hospital, where I have a complex IBD clinic. So in fact, in genetics and IBD, we've known about for about 40 years, since really seminal twin studies. And what those twin studies showed was that if you're an identical twin, it's much more likely that both of you will end up with IBD if one of you has it, compared to if you're a non-identical twin. And the assumption is that given that twins grow up in the same household and they're exposed to the same environmental influences, this is an indicator that there must be something genetic going on, because identical twins share exactly the same DNA. Non-identical twins, it's only 50% of their DNA. And that realization spawned about 40 years worth of effort to try and characterize exactly what that genetic risk was. And this really began with what are so-called linkage studies. In linkage studies, you identify someone who has IBD and maybe their parents or two other members of their family. You pick your favorite bit of DNA and you sequence that to then see whether or not there are genetic variants that co-segregate with the disease and aren't present in people who don't have the disease. And the only thing you need to know about the linkage era was that it was phenomenally unsuccessful. And actually, over over sort of 20 years of linkage genetics, we really didn't learn very much apart from the finding of NOD2. The whole of the genetics era was turned on its head in around 2006 when we first were able to do what's called genome-wide association studies. And that meant for the first time, we didn't have to get lucky and pick a bit of DNA that we thought might be interesting. We could actually screen all of our DNA for genetic variants that might be commoner in people with disease. Now, when you do genome-wide association studies, you generate what are called Manhattan plots. Each of these regions you can see here are different chromosomes. And what you want to see are these towers. And these towers are where there are genetic associations. This has been thought to look a bit like the skyline of Manhattan, hence the name Manhattan plot. And it was pretty clear from the beginning that Crohn's was going to be a much more fertile area for hunting for genetics than maybe, say, bipolar disorder or hypertension. One thing we realized was that actually the key bit about doing genome-wide association studies is numbers of samples. The more you have, the more you will find. And actually, by working together with colleagues around the world, very quickly we were able to increase the number of associations we found up to around 30, which meant that already within a year or two of having GWAS, we vastly outstripped the number of associations that were found through linkage. And that pattern has only really increased. And that's been made possible in part by the fact that we can now do cheaper genotyping using something called the immunochip. And that enabled us to really extend to cohorts from all over the world. And this has really been very much an international effort now. And even by 2011, 2012, we've now got up to well over 100 regions in our DNA that we know are directly implicated in the pathogenesis of inflammatory bowel disease. And we started to learn interesting things about different populations. So, for example, in European populations, one of the biggest risk factors is this gene NOD2. But in fact, that risk factor is not seen at all in people of East Asian background, whereas in those populations, other genes are much more important. We've also, since doing genome sequencing has become cheaper and cheaper, now started to be able to find rare coding variants within specific genes. So we're not just going after the commoner variants, we're now able to get the rarer variants. And this is giving us a much more detailed picture of what the genetic risk of conditions like inflammatory bowel disease looks like. And we're even now starting to move beyond simple susceptibility or risk to start to understand whether there might be a genetic contribution to whether or not you respond to a particular treatment, for example, or whether or not your Crohn's disease or ulcerative colitis behaves in a more aggressive fashion. So why have we done all of this? And I should say that this has been a hugely successful enterprise, and we now have well over 300 regions in our DNA that we know are directly involved in IBD. Well, it's not just a stamp collecting exercise, because actually one of the things we've now realized is that if your drug that's going into a new clinical study, so a brand new drug being tested in IBD for the first time, if that drug hits a pathway that's implicated by genetics, it's much more likely to work. Remarkably, actually only 1 in 10 drugs that go into these sorts of studies ever actually end up getting approved. And the majority of those failures are simply because that drug doesn't make people with Crohn's disease or ulcerative colitis better. We know that that chance is at least double if you're going after a pathway that's implicated by genetics. And in fact, more recent studies have suggested it might even be 6 or 7-fold. So you can take your 10% success rate and turn it into something like a 60 or 70% success rate. But we have a really big problem. And that problem is that most of these genetic associations do not lie in the coding sequence of genes. So they're not having predictable effects on protein structure or function. In fact, the vast majority lie in what are called enhancers. And enhancers are essentially the volume dials in your genome. They are the things that turn up or turn down the amount of genes that are being made. So they determine the where, when, and by how much individual genes are being turned on. And don't forget that actually every cell in your body has the same DNA, but what makes a cell in the back of your eye different to a cell in your gut and different to a cell in your skin are the genes that are being turned on. And that's all being driven by the activity of enhancers. So while there is an amazing potential for genetics to tell us a bit more about why people develop IBD and highlight the pathways that may be going wrong, at the moment, most of that potential is unrealized. So where does that leave us? Well, it leaves us with very long lists of genetic associations where we know the chromosome where the association is on, we know the site on the chromosome, we know some of the genetic variants. And people have slightly unkindly said that, unless we understand the biology, this is about as interesting as reading the telephone directory, for those of you old enough to remember what a telephone directory looks like. And you can kind of understand that because here's a zoomed in version of that. So we have the chromosome, we have the site on the chromosome and some of the genetic variants. But after that, it becomes guesswork. So these are what we've decided are the key genes that might be implicated. And so you think, well, how simple, this must be relatively straightforward. Well, unfortunately, it's not. So here's a genetic association where we've highlighted two genes. It may not be both of those, it might be one or other. But then you say, well, how many other genes are there at that site? And the answer is there are another 63. So how confident are we that we definitely got the right ones? And the answer is often not that confident. But what about these? These are perhaps even harder. Here is your robust genetic association. But actually, when you ask the question, how many genes are present? The answer is none. And these are what used to be called gene deserts. So they're big bits of our DNA in which there are no obvious coding genes. And at this point, people have slightly despaired and thought, well, actually, maybe genetics is not going to be that useful afterwards, after all. Actually, I don't despair, and the people in my lab don't despair, because we like to see this much more like a treasure map, because we've basically got an X marks the spot. We know exactly where something important is going on that's directly involved in why people get IBD. It's just that we're going to actually have to go and do some digging to find out what that treasure might be. And that's exactly the work we do in my lab at the Francis Crick Institute. So what can we learn from genetics? Well, this is a story that we published last year. This is an interesting region. It would have been one of those regions where there would have been a zero. So it's a gene desert. There's no obvious genes there. And what people have done so far when you find these associations is typically ignore them because it's very difficult to make up a hypothesis as to what might be going on when there's not even a gene there that you can pin this on. The interesting thing about this association, which is on chromosome 21, is it doesn't just increase your risk of both forms of IBD, it actually increases your risk of 4 or 5 different inflammatory diseases. So this is that association. The dots here are all genetic variants, and the further they are up the y-axis, the more significant they are. And you can see that at the site of the association here, there are no genes directly underneath. Now, there are some genes nearby. So we figured that this must be one of those volume dials. It must be a controller in some way of some nearby gene. And in fact, we can go looking for this, and the exact details of the method don't matter, but we essentially have a way to identify whether or not there is a volume dial that's turned on at this site. And interestingly, what we found was that there was, and it was very strongly turned on, but only in one type of immune cell, so-called monocytes or macrophages. And these cells are known to be really important in inflammatory bowel disease. They produce many of the inflammatory chemicals that we block with our best treatments. The other thing we know is that although I've portrayed the DNA here as a nice straight line, in fact, it's not a nice straight line within the nucleus. There are bits that are looping from this site, perhaps to other regions. And in fact, what we know here, what we could show was that this region, although everyone thought it was probably going to be one of these two genes that are nearby, in fact, this is looping to an entirely different gene, a gene called ETS2, which is located quite a long way away. And what we could show is that if you carry the allele that increases your chance of getting Crohn's and ulcerative colitis and all these other diseases, it typically led you to have higher levels of this gene ETS2, specifically within your monocytes and macrophages. So the obvious question then was, what on earth is this gene doing? And at the time we began this work, no one knew what ETS2 was doing in macrophages. And actually, sometimes biology is quite simple. And if you want to figure out what a gene does, the easy way to do it is just to turn it off and see what happens. It's a bit like finding a switch in your house and wanting to know what goes on. And if you flick it off and the lights go off, then you know that that's what that switch did. So using a method called CRISPR, we could actually delete this ETS2 gene in these macrophages and then wanted to see what happens. And remarkably, what happens was that the macrophages lose their ability to do almost everything inflammatory. So they stop being able to make inflammatory chemicals. They stop being able to gobble up bacteria. They stop being able to make what are called reactive oxygen species that lead to a lot of the tissue damage in IBD. And in fact, if you can turn something off, the other thing you can do is turn it on. So you can take resting macrophages that are not in any way activated, and we can just turn on this one gene and see what happens. And remarkably, what happened was that all of those pathways, all of those diverse pathways that are involved in inflammation, all of them turned on just by turning on this one gene. And in fact, people have previously published lists of genes that sort of characterize the inflammatory macrophages that you find in the gut of diseases like Crohn's disease. And the vast majority of these genes turn on significantly when you turn on ETS2. So just to put that in another way, what that means is you can take a resting macrophage, you can turn on just one gene, and that macrophage ends up looking like it's just come out of the gut of someone with Crohn's disease. And among the genes that turn on very highly are actually lots of the inflammatory chemicals that we target with our best treatments, things like TNF or IL-23. So when we do genetics, the purpose of this is not just simply to identify interesting genetic variants. The hope is that with enough success and enough associations, we might be able to distill this first down into genes, but then actually also into pathways. And you might expect that lots of associations will be seen within the same pathway. And actually, again, the methods don't matter too much, but what we were able to do, having defined this pathway, was say, let's take all of the genetic associations that we know of with inflammatory bowel disease. How many of those are enriched within this new pathway we've discovered? And it turns out that this pathway was in fact more enriched for the genetics of IBD than anything that's been described before. So starting with a gene desert, we've not only uncovered the sort of critical gene that turns macrophages on and makes them inflammatory, but we found one of the key central pathways that actually is responsible for when people develop IBD. And the thing is, we've known for some time that macrophage inflammation is really important. We know about that for a couple of reasons. Principally, we know about it because some of our best drugs work by blocking the inflammatory chemicals that macrophages principally make, whether that's anti-TNF or blocking IL-23. And the thing is, we know that when we do that in patients, it works really well in maybe 30 or 40% of people, but a lot of people, it doesn't work as well as we want it to. And there's now increasing realization that maybe if you block two of these chemicals, maybe IL-23 and TNF, actually that works better. And of course, gastroenterologists are simple people. If two is better than one, then at some point they'll realize three will probably be better than two and four might be better than three. And I think what we'll eventually get to is saying that if we really want to have a really effective medicine, what we really need to do is actually block lots of these inflammatory chemicals. Now, that would be unaffordable for any healthcare system if we were trying to use individual drugs against each of these. But it might be possible if we can target the thing that actually switches these macrophages on to become inflammatory in the first place. And we think that's what ETS2 does. So could this provide us a therapeutic opportunity for time in the future? Well, there is no off switch within the ETS2 protein, but actually using a large compendium of drug signatures, so these are genes that change when you put on particular types of drugs, we asked the question of, are any of these signatures looking like they might be acting by turning off ETS2? And the answer was quite a few of them did. Some of the ones that did are actually already drugs that we use, like things like JAK inhibitors. But actually, the commonest class was so-called MEK inhibitors. And MEK inhibitors are really interesting because they're already used in people. They're used for cancer treatment, they're used for some neurological disorders, but they're not used for anything inflammatory. So faced with this information, what we then decided to do was take our inflammatory macrophages in the lab, put on a MEK inhibitor and see what happens. And sure enough, this very nicely turns off lots of the inflammatory processes, as you might predict, by blocking ETS2. And in fact, we can do one step better than that because we can take biopsies from people who have active IBD before they start any treatment. Those can be cultured for up to 24 hours in the lab, and we can actually put drugs into the media that the biopsies are in. And when we do that, we find that actually these MEK inhibitors are capable of downregulating lots of inflammatory proteins, including TNF and IL-23, and a whole load of others as well. And in fact, at a gene expression level, we could show that this is nicely turning down lots of the genes that contribute to inflammation. So we think this is really promising. Before any of you who have IBD go to your gastroenterologist and ask for treatment with MEK inhibitors, one of the things we know about these drugs is they can't be taken for very long periods of time because they do have some side effects. And those side effects are because the target, MEK, does things in other tissues that you don't want to block. We want to target it in macrophages, but not everything else. And so actually work that we're now doing in the lab is to try and specifically deliver MEK inhibitors to macrophages using some next generation technologies that we're working on. So hopefully this will be the beginning of a program whereby we can actually start to develop better treatments for people with IBD in the future, because we fundamentally now understand what some of the key pathways are. So this was a piece of work that was published about just over a year and a half ago. And in fact, it led to quite a lot of attention. You may have seen some of this either on the BBC website or on the front page of The Guardian, for example. But it's also led to an awful lot of attention from around the world, with lots of IBD researchers thinking that this actually may be a real breakthrough in terms of both our understanding of disease and potentially finding out how to treat it in the future. So that's all I wanted to talk to you about. I'll finish with that slide, which is just acknowledging all the people who've specifically contributed to that work. And I'm very happy also to take some questions at the end. Thank you.
Thanks, Professor Lee. It sounds like you've made sort of incredible advancements in the last, 4 or 5 years. So very exciting to see what the future holds. And this is a great opportunity now for us to hand over to Tom Hough, who is the Volunteer Development Officer at Crohn's and Colitis UK and has been diagnosed with Crohn's for some 14 years. Tom is going to now share with us his own experiences of living with Crohn's. So it's over to you, Tom.
Thank you, Nicole, and thank you, James. That's fascinating. It was great from a Crohn's patient when it came out on the news a couple of years ago, and even working at a charity, we were all going very excited for that development. So it was great to see other people have taken it forward and that. So yeah, as Nicole just said, I am been diagnosed with Crohn's disease for 14 years now, but I do also work for Crohn's and Colitis UK. So I'll briefly touch upon who they are, just in case people aren't aware of them, but also aware that I imagine there might be a few people with the condition on the call as well, so might be already familiar. So I'll just put in the chat the link to our website for future reference. So Crohn's and Colitis UK are the leading UK charity for those living with Crohn's disease, ulcerative colitis, and microscopic colitis. And currently, we estimate somewhere around 500,000 people, or 500,000 people, are living with those conditions in the UK. And each year, around 25,000 people get diagnosed with those life-changing conditions. So that's about 1 in 120 people in the UK currently. Obviously, that's just an estimate, but that's the closest we've got at the moment. Very recently, we've launched our next 10-year strategy, and our 4 pillars around that are transforming or looking to transform everyone's understanding of Crohn's and colitis, so raising awareness, so it's no longer a hidden disability or condition in the wider general population. We aim to unite researchers, patients, and healthcare professionals to set out a roadmap to ultimately find a cure and improve the lives of people through the science. We also look to drive improvements in diagnosis, treatments, and the care, so improve things in the NHS. And we ultimately also wish to support people to live well through our online information on online events and community events that we run. Again, I'm mindful that people on the call might be familiar with what IBD is, but I will just briefly touch upon what it is, or very briefly, not being a science person on the call, but a patient. But yeah, so it's inflammation of the gut from the mouth to the anus for Crohn's disease, and for ulcerative colitis, it's inflammation of the large bowel. Many people are diagnosed under the age of 30. I was diagnosed when I was 24, and that's pretty typically average for the kind of peak of diagnosis. So it can be described as a kind of a young person's disease, but you can get it at any point or diagnosed at any point in your life. Currently, there is no cure, but symptoms can be tried to be managed through medication and ultimately sometimes surgery, which was the case with myself. The kind of main symptoms are pain, fatigue, diarrhea, constipation, sickness, bloating, and weight loss. Some people may have all of them, some may have a few of them. And I definitely had a few of them, and still do have a few of them to kind of live with on a day-to-day basis. A way to get diagnosed with it, or the ideal way to get diagnosed with it, is having blood tests to check for inflammation if you have any concerns, or issues with your bowels, to go get them checked by your GP. They can also do calprotectin tests on your poo to see all the inflammation levels in there, and also through endoscopy and colonoscopy. And also there is a very useful symptom checker on our website that people can use if they have any concerns or about their bowels and may think they may have Crohn's or colitis and need some sort of checker to go and see. It doesn't obviously diagnose you with a symptom checker, but it does help to kind of reinforce when you have those conversations with GPs to potentially need to look into further tests or investigations. But coming back to myself and my patient story, it was far from ideal. Around 15 years ago, around this sort of time, actually, I started to have bowel symptoms of constipation, pain, bloating, and sickness that wouldn't really go away. They'll be there for a couple of weeks and then relapse for a bit and then come back a bit and then go away a bit. So on and off. And I just wrote it off as stomach issues or just something I ate or that, tried to manage it through that way. I started to go to the GPs, try to see what was wrong, because obviously after a few rounds of that, it was obviously something to look into. I was first dismissed isn't the right word, but they just said it was a stomach bug, nothing to worry about. So I carried on with my life, trying to work through that. But ever increasingly, those bouts of constipation, pain, and bloating got more and more severe, and would there be less downtime in between them. So I went back to the GPs again. I saw a different GP this time, just because of the urgency of needing to see someone. They diagnosed me with irritable bowel syndrome, which people might be familiar with. It's kind of an umbrella term for lots of bowel issues that people can have. And they prescribed me with peppermint pills to see if that could help. It didn't help. I always joke saying all it did was made my stick taste like peppermint, and that. So it still continued on with the issues. So then I went back to the GP again, saw a different GP this time. They didn't think it was IBS. They thought it was something else, and I needed to be referred to a gastro, and that was a 2-month wait to see a gastro. So throughout waiting to see a gastro, continued on with the pain, bloating, sickness, struggling to eat. I was losing quite a lot of weight by then. By diagnosis, I'd lost maybe about 2 stone in weight because I was struggling to eat while still trying to hold down a job. Ultimately, when I saw the gastro, they just referred me to a colonoscopy, and again, I had to wait for another 2 months for that. And by that time, I went back to the GP and said, I just can't take this anymore. Please sign me off work. Nothing's working. Nothing's helping. I didn't know how I was going to get or survive the next 2 months waiting for that colonoscopy. And they said, actually, I'm not going to sign. I can sign you off work, but what I'm actually going to do is I'm going to refer you to a surgeon at your local hospital, because you look very unwell. From the last time I've seen you, you look like a ghost. Your blood test looks like you're bleeding internally, potentially, from what I was describing. Well, my stools up, my poo has gone black. That was the symptoms at the time. So he was very sure that I was bleeding internally. And so they could refer you to a gastro, but they will only refer you to a surgeon, because I think they have to look at you. So I went to my local hospital the same day. They did one X-ray and diagnosed me pretty well straight away. That said, we think you've got Crohn's disease. There's a really bad stricture in in the joining of your large and small bowel, and that needs to be removed. You can go home today if you wish to, but ultimately you will need to come back and have that part of your bowel removed. So I had that removed 14 years ago, towards the end of November. So this time 14 years ago, I was in hospital recovering from part of my bowel being removed and being diagnosed with this rather unknown condition at the time. I didn't know what Crohn's disease was, but I was just grateful that they were able to improve my health, and I got discharged and was kind of in a very negative headspace from what I've been through in that previous year and having a part of my bowel removed. But my health did improve from the surgery, and I started to be more active again, was able to start doing marathons and cycling challenges and climb Kilimanjaro. So very, very active for 3 to 4 years afterwards. And the only main issue I had was the increased frequency of going to the toilet, which probably related to having a shorter bowel due to the surgery and that. So my story may seem extreme, but even 15 or 14 years later, the stats still tell us that 1 in 4 people are just like me, where it takes them over a year to get diagnosed with the conditions, and just like me, 1 in 7 people get diagnosis only via being admitted through an emergency into A&E. So it's still a struggle for people to get diagnosed, hence why the charity is trying to invest in trying to drive improvements in diagnosis and treatment pathways, and obviously in research that people like James Lee are doing. So since then, so that was about 2017, my condition got worse again with other issues, the perianal issues this time, and I've had about 12 surgeries related to that due to complex fistula. So that's been on and off for the last 7 or so years, and I very recently had a EUA, so investigation under anaesthetic, at the end of October. So it's very still new to me, or not new. It's still very a thing in my life, and it obviously always will be, with it being a chronic illness. And oh yeah, and I forget, I've made notes here, but I also had another part of my bowel removed earlier in the year. So yeah, I'm kind of forgetting what's happened to me this year. It's been quite a big year for me. So yeah, that is another stricture in the same sort of area. So I have the type of Crohn's that seems to just have strictures in that area. But yeah, that all went well. And yeah, the current symptoms I struggle with are fatigue and pain as well. But yes, that's me in a very rushed nutshell.
Well, thanks, Tom, for sharing your journey. I think we're all mindful that you've been through a heck of a lot in the last 14 years, and I hope now you can see some sort of positivity and start climbing Kilimanjaro again in the not too distant future. Also the Crohn's and colitis, obviously we've heard about them, but they're an amazing resource. So I'm sure really would be really helpful and useful for many people on this call. And we put it in the group chat if anyone wants to reach after you afterwards. But thank you for sharing your very personal journey and being so open. So at this point, I'm going to hand over to Gideon Schwartz, our Outreach and Education Executive, who is going to lead on the Q&A, and I'm sure there'll be some questions for yourself and some questions for Professor Lee. So thank you very much.
Thank you, Nicole. So if you have any questions, feel free to put it in the chat, raise your virtual Zoom hands. I'll get through. So we had 2 related questions sent to me while you were talking, Professor Lee, in which it was saying that what determines whether you actually get Crohn's or colitis and how responsive you are to the MAB drug? I mean, it seems to not be just if you have this variant, you will definitely be it. And linked to that is, what are the likelihoods of someone that has Crohn's passing it on to their children?
Yeah, so those are good questions. So essentially Crohn's happens when you have a sort of genetic risk, and the genetic risk is not just like like sort of BRCA, for example, where you have single damaging mutations, but actually it's a constellation of mutations across your genome that all increase the risk in a small way. And that genetic risk then has to interact with some sort of environmental trigger. And we're seeing that the rates of diseases like Crohn's and ulcerative colitis have gone up hugely in the last sort of 50 years or so. Of course, the genetics hasn't really changed in that time. So that has to be the abundance of the environmental trigger. And people have linked it really to westernization. Now, whether it's components of the Western diet, or whether it's pollution or microplastics or stress, or there's lots of potential things. We don't fully understand exactly what it is, and it may not be exactly the same for everybody. In terms of what determines whether people will respond or not, we're still trying to do work to try and understand, not just for the drugs we're developing, but for all drugs, what are the things that determine whether people will respond well, or whether, or they might get side effects, and that's other work that's ongoing. And as I say, one of the big challenges in IBD is, although we do have an understanding of what's going wrong at a very high level, we really don't understand the mechanisms very well. And the evidence for that is very much that the drugs that are going into clinical development more often fail than succeed. And that's not because the drug is not doing what you want it to do at a biochemical level, for example. It's just that doing that in someone with Crohn's makes no difference.
Very, very interesting. Trying to read this question from Matthew. If the potential drug targeting ETS2 that significantly reduced the ability to fight infections, is it due to reduced activity of macrophages or just the drug restoring the patient's overall macrophage to normal levels? If it's almost on the quality or quantity is almost there.
Yeah, no, that's a good question. So when we found this, this sort of central pathway that seems to be going wrong, one of the questions was, well, why do we even have it? Why is it there? And why are the variants that increase this pathway so common? And we actually went looking. We've done collaborations with other people who I work with at the Crick who study ancient DNA and sort of, do genome sequencing on Neanderthals and various other ancient humans. And actually, to our surprise, we found out that this genetic variant is incredibly old. So it's at least 500,000 years old. It's potentially even up to 1 million years old. And what that told us immediately was this must be doing something else that in a different context is beneficial. And actually further work in the lab has shown that this response is useful physiologically for when you first see bacteria. So when you get inoculated with the bacteria, the ability to turn on ETS2 in your macrophages is one of the things that enables you to rev them up, enables you to clear that bacteria very quickly. And what that led us to realize is that actually what we need to do is turn this down, not turn this off. Because if you turn it off altogether, you might then leave people unable to adequately fight bacterial infection. So the work we're doing in the lab at the moment is trying to find the right way to turn it down so that exactly as you say in the question, what we're doing is taking the overactive macrophages and reverting them back to normal macrophages without compromising the ability of those macrophages to respond if, for example, there was a bacterial infection.
Amazing. And then the question from Ayala, if IBD drugs often face challenges with targeted delivery, what approach do you see as promising for achieving a localized therapeutic effect in the gut with genetics medicine?
So IBD, both Crohn's and ulcerative colitis, are in fact systemic diseases. Although they principally affect the gut, they can also affect the joints, the skin, the eyes. Tom's talked about the fatigue he experiences, which again is a sort of systemic effect. And so we don't want to just limit the effect of any drug just to the gut. The drugs that we're developing, what we're trying to do is deliver them specifically to macrophages. And the way we're doing that is taking advantage of a technology that has recently been used in cancer treatment, whereby you can have an antibody, and the antibody really serves as your delivery system. So you can attach the drug you're interested in to the back of the antibody. The antibody only recognizes a molecule that's expressed on the surface of these inflammatory macrophages. And by taking that drug to the macrophages, by virtue of using the antibody as a sort of delivery tool, that enables that to be then taken up by the macrophages, the drug cleaved, and then that drug then gets released inside the macrophages and then shouldn't have all the side effects that it might have if it were able to get into other tissues. So this is the sort of technology that we're working on at the moment.
There was a question for Thomas. What advice would you give to someone that thinks they might have Crohn's or colitis? Is there somewhere you seem to have almost a diagnostic odyssey, to use a term from rare diseases, where you were moving in and probably nothing, or it's just IBD, ending with obviously serious things. So what would they say to a GP? How would you, almost if you were to give advice to your 14 years ago self of what he should have said in his first GP appointment?
Well, the obvious thing that could jump to mind when I read the question was that I would ask them, do they think it's Crohn's disease or ulcerative colitis or IBD? Because without asking that, you won't know if the GP's ruled it out or even thought of it, because it's not, as shown in my story, it's not always the first thing that jumps to GP's mind. They'll obviously do the most obvious and investigations and that sort of thing. And there is also a symptom checker on the Crohn's and colitis website, which might be useful for people to complete, and you can print off the results to that and take it to your GP to go, I've completed this symptom checker, and they're potentially suggesting or could be suggesting that further tests are required, and knowing what the tests are that can also help towards a diagnosis can also be part of your toolkit when going into speaking to the GP and about those kind of blood tests that I mentioned, looking for inflammation or a calprotectin test. More GPs are getting more knowledgeable about IBD and the diagnosis pathway, but again, part of our charity's work is to improve that primary education. But yeah, as a patient, we obviously have to arm ourselves with knowledge as well. And I just wish I had that knowledge 14 years ago, and we would have hopefully had a more smoother diagnosis pathway. But I don't know if James has got any, James Lee's got anything to add into that. I know you're a gastro, so you might have more.
No, I mean, I think GPs are getting better. And I think the ease of things like doing the stool test for calprotectin has made it much more easy, because one of the things we know is that irritable bowel syndrome, which I know you were diagnosed with, doesn't increase your calprotectin. So it's now, there is a quick test. And one of the big challenges of diagnosing something like Crohn's is that many of the symptoms do look a lot like irritable bowel syndrome. And if you take, 10 patients who are going to the GP with these sorts of symptoms, maybe 9 of them will have IBS and 1 of them might have Crohn's. So it is hard for GPs sometimes, but there are better tests and people are now getting more awareness, and that's an awful lot of the work that Crohn's and Colitis UK are doing.
Thank you. And then merging a couple of questions from Nick of, if a Crohn's patient had genetic testing, would results show a genetic deficiency, or do some Crohn's patients not have any genetic predispositions? And then similarly, would everyone with the genetic predisposition develop Crohn's? And if not, why not?
Yeah, so those are good questions. So I'll take the second part first. So not everyone who has the predisposition will develop Crohn's disease. And the way I like to try and explain this to patients is it's a bit like the firing of a gun. So the genetics loads the bullets into the gun, and then the environmental trigger pulls the trigger, and then the gun fires. If your gun has no bullets in it because you have no genetic risk, you're not going to get Crohn's disease, even if you're exposed to the environmental trigger that could set it off in someone else. Conversely, if your gun is fully loaded, but you never see the trigger that sets it off, you're also not going to get Crohn's disease because you need to see the environmental trigger as well. So we know that for anyone who has Crohn's disease, their first degree relatives have somewhere between a 5 and 10% chance of developing Crohn's disease in their lifetimes. So to reassure people, that means there's 90% chance they won't develop it. But obviously, if people do get symptoms that are suggestive of this, they need to go and get investigations done.
And then a follow-up question, which is for a PhD student studying colitis-associated bowel cancer. So one of the more expert people in the audience, I'd say. As many of the apparent downstream targets of ETS2 are involved in angiogenesis, such as MMPs, IL-8, and TNF, have you seen any effects on angiogenesis when knocking down overexpression of the gene? Or do you predict this gene could be involved in development of colitis-associated cancers? And just as a rough explanation of what that question means might be helpful as well to me, at the very least.
Yeah, so that's so angiogenesis is the development of new blood vessels. And so the question is essentially, could the inflammation that is ETS2 is driving contribute to the development of new blood vessels, which actually is a feature that we sometimes see in active IBD? And the answer is yes, it could. We haven't shown that definitively ourselves, but we know that development of new blood vessels and expansion of existing blood vessels is a feature of what we see in IBD. In terms of the second part to this is, could this be involved in colitis-associated cancer? We know that in patients who have chronic inflammation in their colons, they are at a higher risk of developing bowel cancer. And so patients with IBD, after a period of time, will start to be screened on a regular basis for this. We know that a lot of the pathways that are involved in that process of the development of cancer are due to inflammation, and ETS2 is one of the drivers of this. So we suspect it probably is. And again, the best thing that you can do to reduce your chance of getting bowel cancer in the context of ulcerative colitis or Crohn's disease is to actually get the inflammation under control. And that's exactly what we're trying to find new ways to do.
Brilliant. Do we know the proportion of people with Crohn's that do have this ETS2 gene mutation?
Yeah, so it's high. So about 95% of patients with Crohn's will carry 1 or 2 copies of this mutation. So it's incredibly common. It's also quite common in people who don't develop Crohn's. And the whole point of the way in which you develop IBD is that you need hits in multiple different pathways. It's not enough just to have a single mutation, but this is certainly one of the main mutations within this inflammatory macrophage pathway.
A couple more, I think. Do we have any more clues as to what the environmental triggers are beyond the catch-all westernization?
Yeah. So we do. I mean, one of the things, studying genetics is great because you can get to sort of mechanism and you can start to do all the sort of studies that we've done to figure out exactly what's going on. Studying environmental risk factors, which you do with things like epidemiology, is much less precise. And you often get lots of things that are like, westernization, diet, pollution, everything else, but you never get causation. You never get the sort of smoking gun to say that this is exactly what caused it. One of the pieces of work that we're doing in the lab at the moment is that we seem to have found a pathway that exists in people that actually essentially is the braking mechanism that seems to switch ETS2 off. And that's interesting because the braking mechanism seems to depend, at least in part, on diet. So we're doing work at the moment in the lab to try and understand exactly what this is, because the implication for that would be, we know that a Western diet is the biggest risk factor, maybe apart from smoking in some cases. And everyone has always assumed that it must be something bad in a Western diet, whether it's ultra-processed food or refined sugar or whatever it might be. Another explanation, and these things are not mutually exclusive, is that it might be that there was something that we used to eat, in times gone past, but which we don't eat anymore, but which is protective. So it's the sort of absence of a protective factor rather than the presence of a trigger that's setting it off. And that's important because if that is the case, even if it's only part of the story, then that's something that might be fixable in a way that it's much harder to try and fix ultra-processed food or refined sugar or whatever else it might be. If it's just a question of supplementing something back into the diet, then that's something that we might be able to do in the future. So that's ongoing work in the lab at the moment.
Thanks, Raj. And one final question, which Nicole asked in the chat, a couple have also found a message, which is, as we said, anecdotally, we seem in the Jewish community to know a lot of people affected by Crohn's and colitis. Is there any evidence or any evidence or specific predisposition that we know linking within Jewish community and also other general ethnicities towards Crohn's and colitis?
Yeah, so we know that if you take the genetic, the 300 or so genetic variants that we know of for IBD at the moment, many of those are much commoner in Jewish populations. And that's almost certainly the main contributor to why the rates are much higher in Jewish populations than in some other populations. So I think that's probably the best explanation. We don't think it's that the environmental trigger, whatever it might be, is something that's particularly more associated with a sort of Jewish lifestyle. But it's almost certainly that the genetic risk is much higher in Jewish populations.
Thank you very, very much.